Hello and welcome to BiochemSerye, or the Biochemistry Lesson Series. This series is entitled The Basics of Chemistry. It is important that we review some basic concepts of chemistry before diving straight onto studying the much more complicated world of biochemistry. This series is composed of 6 lessons. Please feel free to subscribe to be updated of the latest lesson installments for this series. The different lessons for this series is enumerated as follows. The first lesson involves the basis of chemistry. That is, the different elements or atoms which can be found in the periodic table. This is followed by Lesson 2, which will discuss the chemistry of molecular bonds or how atoms are held together. Thirdly, we will investigate the molecular structure and its different components and concepts. Next is we will delve into the concept of isomerism. This will then be followed by Lesson 5, which will look into the different molecules that play important roles in biochemistry. And lastly, we will see how different molecules interact with each other in the form of reactions. This video includes only Lessons 1 and 2. The succeeding lessons are in the subsequent videos. Please subscribe and click on the notification bell to be notified of the succeeding lesson uploads. There are 81 stable elements in nature. 15 of these are present in all living things, and a further 8 to 10 are only found in particular organisms. The illustration shows the first half of the periodic table, containing all of the biologically important elements. In addition to physical and chemical data, the periodic table also provides information about the distribution of the elements in the living world and their abundance in the human body. All living cells are composed of a set of elements. They are around 92 naturally occurring elements. But in biological organisms, there are only a number of elements that play important roles. The major constituents of most biomolecules, comprising 96.5% of all the atoms in an organism, are carbon, oxygen, hydrogen, and nitrogen. All living cells are composed of a set of elements. Hydrogen is the most abundant element, comprising of almost 50% of all the atoms in a biological organism. Carbon and oxygen atoms come next, each making almost 25% of all atoms. Nitrogen comes a far fourth. Other elements, though minute in amount, play crucial roles in biochemical processes. Phosphate is a component of nucleic acids. Calcium plays a key role in innumerable biologic processes. Other constituents include iron, iodine, potassium, sodium, chloride, and magnesium. These elements are usually encountered in clinical practice in patients with various diseases. All living cells are composed of a set of elements. As mentioned, the major constituents of most biomolecules are carbon, oxygen, hydrogen, and nitrogen. The most abundant substance of the living cell is water, which accounts for about 70% of a cell's weight. Most intracellular reactions occur in aqueous environment. A second biologically important group of elements, which together represent only about 0.5% of the body mass, are present almost exclusively in the form of inorganic ions. This group includes the alkali metals sodium and potassium, and the alkaline earth metals magnesium and calcium. The halogen chlorine is also always ionized in the cell. All other elements important for life are present in such small quantities that they are referred to as trace elements. These include transition metals such as iron, zinc, copper, cobalt, and manganese. A few nonmetals, such as iodine and selenium, can also be classed as essential trace elements. The chemical properties of atoms and the types of bond they form with each other are determined by their electron shells. The possible states of electrons are called orbitals. These are indicated by what is known as the principal quantum number and by a letter S, P, or D. The orbitals are filled one by one as the number of electrons increases. Each orbital can hold a maximum of 2 electrons, which must have oppositely directed spins. This figure shows the distribution of the electrons among the orbitals for each of the elements. For example, the 6 electrons of carbon shown here occupy the 1s orbital, the 2s orbital, and 2p orbitals. A filled 1s orbital has the same electron configuration as the noble gas helium, or He. This region of the electron shell of carbon is therefore abbreviated as He. Below this, the numbers of electrons in each of the other filled orbitals, the 2s and 2p in the case of carbon, are shown on the right margin. For example, the electron shell of chlorine, shown here, consists of that of neon, Ne, and 7 additional electrons in 3s and 3p orbitals. In iron, A transition metal of the first series, electrons occupy the 4s orbital even though the 3d orbitals are still partly empty. Many reactions of the transition metals involve empty d orbitals. For example, redox reactions or the formation of complexes with bases. Particularly stable electron arrangements arise when the outermost shell is fully occupied with 8 electrons, also known as the octet rule. This applies, for example, to the noble gases, as well as to ions such as chloride or Cl- and sodium +1. It is only in the cases of hydrogen and helium that 2 electrons are already sufficient to fill the outermost 1s orbital. Stable, covalent bonds between nonmetal atoms are produced when orbitals of the 2 atoms form molecular orbitals that are occupied by 1 electron from each of the atoms, as we previously saw. Thus, the 4 bonding electrons of the carbon atom occupy 2s and 2p atomic orbitals as shown here. The 2s orbital is spherical in shape, while the 3 2p orbitals are shaped like dumbbells, which are arranged along the x, y, and z axes. It might therefore be assumed that carbon atoms should form at least 2 different types of molecular orbital. However, this is not normally the case. The reason is an effect known as orbital hybridization. Combination of the s orbital and the 3p orbitals of carbon gives rise to 4 equivalent tetrahedrally arranged sp3 atomic orbitals or sp3 hybridization. When these overlap with the 1s orbitals of H atoms, 4 equivalent sigma orbitals are formed. For this reason, carbon is capable of forming 4 bonds. That is, it has a valency of 4. Single bonds between nonmetal atoms arise in the same way as the 4 sigma or single bonds in methane or CH4. For example, the hydrogen phosphate ion, HPO4-2, and the ammonium ion, NH4+1, are also tetrahedral in structure, as shown here. A second common type of orbital hybridization involves the 2s orbital and only 2 of the 3 2p orbitals. This process is therefore referred to as sp2 hybridization. We can see here that this will result to 3 equivalent sp2 atomic orbitals in a trigonal configuration. That is, there are 3 equivalent sp2 hybrid orbitals lying in one plane at an angle of 120 degrees to one another. The remaining 2px orbital is oriented perpendicular to this plane. In contrast to their sp3 counterparts, sp2 hybridized atoms form 2 different types of bond when they combine into molecular orbitals as shown here. The 3 sp2 orbitals enter into sigma bonds, as described previously. In addition, the electrons in the 2 2px orbitals, known as pi electrons, combine to give an additional, elongated pi molecular orbital, which is located above and below the plane of the sigma bonds. Bonds of this type are called double bonds. They consist of a sigma bond and a pi bond, and arise only when both of the atoms involved are capable of sp2 hybridization. In contrast to single bonds, double bonds are not freely rotatable, since rotation would distort the pi molecular orbital. Therefore, all of the atoms lie in one plane. In addition, cis and trans isomerism arises in such cases. We will be discussing isomerism later in the succeeding lessons. Double bonds that are common in biomolecules are double bond and C double bond O. C and double bonds are found in aldimines. Many molecules that have several double bonds are much less reactive than might be expected. The reason for this is that the double bonds in these structures cannot be localized unequivocally. Their pi orbitals are not confined to the space between the double bonded atoms, but form a shared, extended pi molecular orbital. Structures with this property are referred to as resonance hybrids, because it is impossible to describe their actual bonding structure using standard formulas. One can either use what are known as resonance structures, that is, idealized configurations in which pi electrons are assigned to specific atoms, or one can use dashed lines to suggest the extent of the delocalized orbitals. Resonance-stabilized systems include carboxylate groups, as in formate, aliphatic hydrocarbons, with conjugated double bonds, such as 1,3-butadiene, and the systems known as aromatic ring systems. The best known aromatic compound is benzene, which has 6 delocalized pi electrons in its ring. Extended resonance systems with 10 or more pi electrons absorb light within the visible spectrum and are therefore colored. This group includes the aliphatic carotenoids, for example, as well as the heme group, in which 18 pi electrons occupy an extended molecular orbital. This concludes the first and second lesson in the Basics of Chemistry lecture series. The next episode is comprised of the third and fourth lessons as shown here. Thank you very much. Thank you for watching this episode in this playlist. Check out the other videos in this playlist and please do subscribe to our channel and click the bell to be notified of new video episodes that will be regularly uploaded.